Hello everyone, my name is Rafael Ives C. Makatugal and I'm going to report about hollow fiber membranes of PCL and PCL graphene as scaffolds with potential to develop in vitro blood-brain barrier models. Abstract. There is a huge interest in developing novel hollow fiber membranes able to modulate neutral differentiation to produce in vitro blood-brain barrier models for biomedical and pharmaceutical research. Due to low cell-inductive properties of polymer used in current blood-brain barrier models, in this work of PCL and composite PCL graphene, membranes were prepared by phase inversions and were characterized in terms of mechanical, electrical, morphological, chemical, and mass transport properties. The presence of graphene in PCL graphene membranes enlarged the pore size and the water flux and presented significantly higher electrical conductive than PCL hollow fibers. A biocompatibility assay showed that PCL graphene hollow fibers significantly increase C6 cells adhesion and differentiation towards astrocytes, which may be attributed to their higher electrical conductivity in comparison to PCL hollow fibers. Introduction. The application of membranes in medical field has gained interest in the last few years and represents one of the most relevant markets for membranes. Among the different biomedical applications, the use of membranes for tissue engineering as scaffolds help to ensure sufficient nutrient transport and to promote cell attachment and proliferation, mimicking psychological vascular networks and engineering tissues in vitro. This approach might serve to produce implantable tissue engineered devices, but also in vitro cell tissue models that recapitulate tissue functionally to screen new pharmaceutical and or to study mechanisms of the progressions of diseases. With the increasing concern of traumatic brain injuries, neurodegenerative diseases, and brain tumors, the progress of new therapies designed for central nervous system illness is crucial for ensuring social and economical sustainability in an aging world. In this line, tissue-engineered models could enable the study of clinically relevant processes such as CNS drug delivery efficient through the blood-brain barrier. The BBB is a dynamic and complex structure of brain capillaries that tightly regulates exchange of nutrients, oxygens, and metabolites between the circulating blood of the extracellular fluids of the nervous tissue in the CNS. The BBB is essential for control of CNS homeostasis and the protection of the nervous tissue from toxins or pathogens. And alterations of this barrier underlie the progression of different neurological disorders. Materials and methods. Preparation of PCL graphene-based nanomaterial hollow fibers. In the present study, instead of laboratory-made graphene oxide or reduced graphene oxide which are widely studied in literature, we decided to explore the feasibility of using commercial graphene or graphite nanopellets produced at a large industrial scale by mechanical exfoliation. The PCL and PCL graphene hollow fibers were fabricated by non-solvent induced phase separation produced by immersion, precipitation using a small dimension spinning setup to process the polymer solution. Polymer solution and bore fluid were simultaneously pumped through a tube in orifice spinneret. Here, as you can see, the formation conditions for the fibers are summarized in Table 1. From each polymer solution, several meters of hollow fiber membranes were prepared, and only homogeneous and reproducible sections were selected for experimental purposes. Psychochemical characterization of a hollow fibers. The structure of morphology of surface and cross-section of PCL and PCL graphene hollow fiber membranes were determined using environmental scanning electron microscopy with a large field detector in low vacuum mode at a voltage of 5 kV. Mechanical, electrical, and flux properties characterization of a hollow fibers. Axial tensile tests of hollow fibers were done using specimens of 50 mm active length with a load cell of capacity of 100 Newton at a constant speed of elongation of 50 mm. The average value of strain properties were obtained from 5 samples of each fiber. The electrical conductivity of the PCL and PCL graphene hollow fiber dry membranes was evaluated by electrical impedance measurements with a programmable automatic RCL meter. Alligator clips were placed at a 0.5 cm along the length of the hollow fibers. Cell cultures. For the cell culture experiment, samples of PCL and PCL graphene hollow fibers were cut into 0.5 centimeters length and placed on the flat bottom 6 well plates. The experimental configuration consists of 2 samples of each hollow fibers and two 10 millimeter diameter glass cover slips as control in each well plate. Last is the statistical analysis. Quantitative cellular experiments were run in duplicate for each cell line and substrate. For the addition, proliferation, and differentiation cellular stages, the total number of cells was counted from 4 micrograms and expressed per square millimeter. To evaluate the differentiation stage of the C6 cell line, morphometric analysis was done to measure the length of the process, defined as the extension from the point where the process emerged from the cell body to the tip of the major process. The number of mature astrocytes, defined as cells with at least 2 processes longer than the cell body, were counted from each image, and percentage of differentiated cells was calculated. Results and discussion. Figures 1 and 2 show representative ESEM images of the cross-section and the outer surface respectively of the PCL and PCL graphene hollow fibers. In Figure 1, cross-section ESEM images PCL and PCL graphene hollow fibers employed to determine the hollow fibers' morphological characteristic as shown in the picture. Next is Figure 2. In Figure 2, outer surface ESEM images of the PCL and PCL graphene hollow fibers show the higher magnification details of external surface of PCL and PCL graphene hollow fibers. From the cross-section of PCL Figure 1A and PCL graphene Figure 1D, hollow fibers with a porous morphology were observed. At a higher magnification, the porous structure of the cross-section of both hollow fibers was asymmetric and contained finger-like cavities or macrovoids, reaching from 2 to 3 of the wall towards the outer surface of and sponge-like structure with micropores towards the internal diameter as shown in Figure 1B, C, E, and F. The ESEM images of the outer surface of PCL Figure 2A and PCL graphene Figure 2C, hollow fibers shown uniform surface morphology. At a higher magnification, Figure 2V and D, the values of mean surface force could be estimated. Here in Table 2, morphological characteristic of the PCL and PCL graphene hollow fibers. Student test statistical analysis of significance considering PCL hollow fibers as a reference. Mean surface force, size, bulk, porosity. The higher viscosity of PCL graphene dope solution with respect to PCL dope solution may have produced a higher shear stress in extrusion needle, causing the expansion of the internal and external diameter of PCL graphene hollow fibers as shown here in the table. Bulk porosity in both hollow fibers was above 80%, similar to the other polymer scaffolds manufactured by phase separation, which guaranteed the uniform supply of nutrients to cultured cells. Here in Table 2, it summarizes the results obtained from the morphological characterization of porosity of the hollow fibers. Here in Figure 3, it shows that Raman spectra of the PCL and PCL graphene hollow fibers and graphene nanomaterial. 4 relevant peaks in the spectra of the commercial graphene were identified. The D band at a 1,325 cm associated with the presence of carbon atoms with sp3 hybridization or structural disorder in the graphene network. The G band at 1,583 cm attributed to the energy of sp2 bonds of C atoms. The D band at 1,615 centimeters superimposed with the G band and related with the edges defects, and finally the 2D band at the 2,635 centimeter associated with the multi-layered character of the graphene. The 1-2D or Ig ratio was 0.35, indicating that the nanomaterial had a 6 to 7 layers, as you can observe here in the table. 3.2 Mechanical, electrical, and flux properties characterization of the hollow fibers. The mechanical stability of polymer scaffolds is important to allow cell adhesion and proliferation. Moreover, the mechanical properties of the hollow fiber must maintain the structure and integrity to ensure the growing and maturation cultured tissues. Here in Table 3, it collects the young modulus yield point, ultimate tensile strength, and elongation at break obtained from axial tensile tests of PCL and PCL graphene hollow fibers. The mechanical properties, particularly elastic modulus, play an important role in cell fate and differentiation. As shown here in Table 3, the presence of graphene in polymer matrix reduces the mechanical properties of the hollow fibers. Here in Figure 4, it illustrates ultra-pure water fluxes for different transmembrane pressures. Ultra-pure water flux at different transmembrane pressure across the PCL is in gray, and the PCL graphene hollow fibers is in black. The bars represent the mean. The student test statistical analysis of significance considering PCL hollow fibers as a reference also. Next is the cell culture's viability on the hollow fibers. In order to test whether the PCL or PCL graphene hollow fibers could be used as a scaffold for cell cultures and be sustainable for producing into vitro blood-brain barrier models, the morphology and the adhesion proliferation and differentiation capacity of the UVEC cells were analyzed on both membranes. The results were obtained compared to the TCP as positive control, as you can observe here in Table 4. Overall, water permanence results were consistent with the ESEM imaging, which showed the high porosity values of the hollow fibers. Here in Figure 5, representative images of confocal microscopy where the density and cell morphology of C6 cells were observed after 24 hours sowing on TCP, PCL, and PCL graphene substrate. Cytochemical techniques have been also used with paludin, which is the green part, direct maker of the actin cytoskeleton, and DAPI, which is the blue part for the cell nucleus. Figure 6, images of confocal microscopy for the study of astrocyte differentiation for 72 hours on TCP, PCL, and PCL graphene substrates. Cells had dramatically reduced their cell and nuclear size in addition to presenting numerous cytoplasmic projections. Last is in Figure 7, mosaic cell culture of UVEC endothelial model over TCP, confocal microscopy images of UVEC cells grown on the surface of PCL or PCL graphene membranes in hollow fiber configuration 24 hours after sowing. In summary, this report reveals that PCL graphene composite hollow fiber is a permissive scaffold that allows C6 cells growth migration and correct differentiation process into astrocytes. The PCL graphene hollow fibers exhibit excellent properties such as electrical conductivity and biocompatibility, making them good candidates for studies of astrocytes cell models. However, the potential toxicity of graphene observed for UVEC cells should be studied profoundly. Furthermore, it will be necessary to develop a bilayer PCL hollow fiber in which graphene must be avoided in the luminal surface of UVEC cells culture and accounting with the PCL graphene external layer for growing C6 cells with the objective of designing functional into vitro blood-brain barrier models where 3D UVEC or C6 co-cultures could be performed. And conclusion. This work refers for the first time fabrication of PCL graphene hollow fibers by phase inversion to be used as a tubular scaffold for cell culture. In particular, the present work aimed to explore the viability of these novel PCL graphene hollow fibers to the future development of the dynamic in vitro blood-brain barrier model. In order to accomplish this goal, the morphological, chemical, and functional properties of PCL and graphene hollow fibers were characterized. The morphological analysis of the hollow fibers found out that both types of membranes presented similar interconnected porosity but relevant differences in the average pore size. Particularly, the values obtained of PCL graphene were significantly higher than the average pore size of the polypropylene hollow fibers used normally in dynamic in vitro blood-brain barrier models. And this provided high water fluxes sufficient to sustain neutral models in vitro. Both PCL and PCL graphene hollow fibers mechanically properties were sufficient to enable their use as scaffolds for cell culture in blood-brain barrier model. That is all, and thank you for listening.